this video, I'll be discussing how does the protist euglena react in ice water. Euglena is a protist that is unicellular and eukaryotic. Unicellular meaning that it only has one cell, and eukaryotic meaning that it has a nucleus. The nucleus is what controls the cell and holds the DNA. In the image, you also see a few other functions of the cell. The mitochondria is the powerhouse of the cell. The endoplasmic reticulum surrounds the nucleus. The Golgi apparatus receives the proteins from the endoplasmic reticulum. The flagella helps it move. The eye spot helps the euglena move towards its food and detect light, and the chloroplast is used to make energy. With this organism having a chloroplast and eye spot, it helps you know that this organism goes through photosynthesis. Photosynthesis is when organisms, usually plants, use sunlight to synthesize food from carbon dioxide and water to produce oxygen and glucose. Euglena can be considered heterotrophic or autotrophic. Autotrophic because it makes its own food when going through photosynthesis. Heterotrophic because it can also consume other organisms as its food. To test out my original question, I had to come up with an experiment. For my experiment, I took 1 drop of euglena and 1 drop of ice-cold water and put them on a slide and then covered the slide with a slide cover. I then placed the slide under the microscope and observed the movement of the euglena compared to what I had observed earlier. Before I did this experiment, I hypothesized that the ice water would slow the euglena down and could possibly not make it move at all. After putting the slide under the microscope, I waited about 5 minutes and observed the euglena again. At this point, most of the euglena had very slim to no movement. When this says preserved euglena, it means the euglena was not alive at the time this picture was taken. In the 10 magnification of the euglena, you cannot see much about the euglena. In my opinion, the 40 magnification preserved slide euglena shows more parts of the euglena. If you look at the picture, you can see the nucleus, which happens to be the darker purple spot in the middle. Also, if you look closely, you can see a small flagella on some of them. This is the euglena in normal conditions, with no ice water added and not preserved. In the 10 magnification, the euglena looks like just a bunch of green little dots. When you see the euglena in 40 magnification, you start to see it a little bit closer. In this magnification, you may even be able to see some of the functions of the cell. In conclusion, I observed that the ice water slowed the euglena down. The euglena seemed to go a little crazy when it came close to another euglena while still exposed to the ice water. After hearing what my peers got and doing some research on euglena, I would say I got a pretty common conclusion. Most of the time, the euglena will indeed slow down when exposed to ice water. The number of euglena when in the ice water seemed to be decreasing. This could be because the sample was not as good or because the ice water was killing them. The longer the euglena was in the water, the slower it moved. To improve this experiment, I could take the temperature of the ice water to make sure it was indeed ice water. I could make sure I get a good sample, and I could do more than one sample to make sure I get the same results for each trial. Another way to improve this experiment is I could have gone to check on the euglena a few minutes after I put it in the ice water and recorded what I observed then. I could have then waited a few hours, maybe even the next day, to see if the euglena was still alive. If it wasn't still alive, then I would definitely know that cold water would kill euglena. Thank you for watching my video. After viewing my video, I hope you have a better understanding of how the euglena works and how all the different functions come together to make the euglena operate as it does. I also hope you understand what the effect ice water has on the euglena.